Welcome to today's BMG LabTech sponsored tutorial. My name is Carl Peters. I am a senior application scientist working out of the US for BMG LabTech. Today, it is my pleasure to introduce Jaime Padrós. Jaime will tell you a little bit about the initial results from a collaboration between BMG and BioAuxilium. At BMG LabTech, our sole focus is on the production and innovation of microplate readers. This allows us to respond to the scientific community and provide them with the reader technology that they need. Jaime will be telling you about the validation of their assays performed on the CLARIOstar Plus. The CLARIOstar Plus is a multi-mode detection reader that is by far BMG's most flexible reader, due in large part to the patented LVF monochromators that are the most sensitive monochromators available. It can also be equipped with a spectrometer for absorbance, as well as highly sensitive filters for advanced assays. CLARIOstar Plus has a number of important features that are useful in the detection of time-resolved fluorescence. These include the highly sensitive filters, but also redshifted PMTs for detection, a free air optical path for best sensitivity, as well as Z-height focus so that you see the optimal signal within the well. The features included in the CLARIOstar Plus equate to an excellent limit of detection in time-resolved fluorescence, which you can see here in this comparison of the CLARIOstar to other similar readers. So without further ado, I would like to introduce Jaime Padrós. Dr. Padrós is the co-founder and president of BioAuxilium Research. Prior to BioAuxilium, Jaime rose to the level of technical project leader at PerkinElmer, where he managed all research activities for the development of drug discovery and research reagents, including Lance Ultra TR-FRET assay kits. He has worked extensively in both assay and reagent development in a number of specialties, including high-throughput screening and biomarker detection. Jaime?
Thank you, Cal, for the introduction and for giving us the opportunity to present this tutorial at this last meeting. It's much appreciated. I will start the presentation by giving you some background about us, BioAuxilium Research. Then I will briefly describe the TR-FRET principle and our TR-FRET technology, Thunder, along with our recent line of TR-FRET cell signaling products. Next, I will present the validation work that we conducted on the CLARIOstar Plus microplate reader with selected Thunder assays, followed by the conclusions. BioAuxilium Research is a dynamic biotech founded in 2013 by former PerkinElmer scientists. We are located in Montreal, Canada at the ADMER Innovation Center, an open access drug discovery hub. Our focus is to design, develop and manufacture cutting-edge immunoassay kits based on our proprietary TR-FRET technology, Thunder. Over the last years, we have licensed hundreds of cellular TR-FRET assay kits to a global reagent manufacturer. In 2018, we pivoted our business model to develop and manufacture our own line of TR-FRET cell signaling assay kits based on our Thunder technology. The first panel of Thunder kits was launched last year. Our goal is to accelerate biomedical research by providing top quality, fully validated, yet affordable TR-FRET assays. We also provide expert, customized service in assay development using a range of technology platforms. Our unique selling point is certainly our expertise and experience. We have a track record of developing hundreds of commercially successful assay kits for drug discovery and basic research. Let's now review what's TR-FRET and how our technology works. The Thunder technology combines FRET with time-resolved measurement of fluorescence. Foster resonance energy transfer, or FRET, is based on the transfer of energy, not fluorescence, between two compatible fluorophores, a donor and an acceptor, when in close proximity. Thunder uses an europium chelate as the donor fluorophore and a small far-red fluorophore as the acceptor. In a typical sandwich immunoassay application, one antibody is labeled with the europium chelate and the second antibody with a far-red fluorophore. When the antibodies bind to the analyte, donor and acceptor fluorophores are brought close together. Excitation of the donor europium with a flash lamp or a laser triggers a transfer of excitation energy to the acceptor, which in turn emits a specific TR-FRET signal at 665 nanometers. The intensity of this fluorescence is proportional to the level of analyte in the sample. Since the transfer of energy occurs only when the donor and acceptor are in close enough proximity, FRET-based assays are homogeneous. That is, they do not require any washing or separation steps to remove distant donor and acceptor partners. Another key advantage of TR-FRET is that data can be expressed and analyzed as the ratio between the fluorescence of the acceptor and the fluorescence of the donor. The use of the ratiometric measurement further increases data reproducibility because it normalizes small pipetting errors and also corrects for some compound artifacts. The key advantage of using a europium chelate as the donor fluorophore is that it emits long-lived fluorescence that lasts for milliseconds, so the specific FRET signal can be measured in a time-resolved manner. How the time-resolved measurement works is shown in this figure. With a TR-FRET compatible plate reader, excitation of the europium chelate by a pulsed light source is followed by the delay and then the signal measurement. This allows short-lived fluorescence to decay before a measurement is made. This eliminates all fluorescent background caused by the sample and plastic microplate, and also by direct excitation of the acceptor, which increases assay performance. Overall, because of their excellent temporal and spectral resolution, the Thunder TR-FRET assays exhibit very low background and high signal-to-background ratios, resulting in higher sensitivity. The TR-FRET detection mode brings additional benefits. Thunder assays have a robust add-incubate-read format that doesn't require any washing. The high resistance of the europium chelate to photobleaching and very long signal stability allows the reading of assays as many times as needed. Thunder assays are easy to automate and miniaturize for screening applications and are also cost-effective. Thunder sets a new benchmark for TR-FRET assay kits for three reasons. First, these kits were designed with quality and performance in mind. They use a pair of donor-acceptor fluorophores showing exceptional spectral compatibility and TR-FRET signal. We conduct extensive testing of labeled antibody pairs using optimized lysis buffers for the highest TR-FRET signal. And we also conduct extensive assay optimization to ensure specificity, reproducibility, and optimal assay performance. In addition, these kits are fully validated. They undergo a stringent validation process using lysates from cells treated with pathway-specific modulators to further confirm target specificity and demonstrate performance in real assay conditions. We include the validation data in the technical data sheets, the most complete in the market. Lastly, we manufacture all kits in-house and we verify lot-to-lot consistency using a functional QC assay where each new lot is compared to the previous lot. In short, Thunder is a cost-effective assay platform that provides easy access to the TR-FRET technology. As part of our product validation efforts, we have benchmarked some of our key assay kits against two competitive TR-FRET platforms. Here, we show comparative data for phospho-AKT pan phosphorylated at serine 473. Specifically, The three different assays were compared side by side for their capacity to detect phospho-AKT pan stimulation upon treatment of MCF7 cells with insulin. Following treatment, cells were lysed and the phosphoprotein measured in the lysate using the corresponding reagents included in each kit. The summarized data clearly show that while the three TR-FRET assays exhibit comparable pharmacology, the Thunder assay shows the highest signal-to-background ratio. Our current product portfolio consists of 68 cellular assay kits and is focused on intracellular kinases involved in key signaling pathways. Our goal, however, is to offer a comprehensive catalog of TR-FRET reagent tools for both cellular and biochemical assays. That's why new cellular kits for the quantification of phosphoproteins in cell lysates and cytokines in cell culture media will be launched soon, along with toolbox reagents for developing biochemical assays. There are several plate readers available on the market capable of measuring TR-FRET assays. However, the choice of a plate reader can have an important impact on the performance of a TR-FRET assay. We have recently started a collaboration project with BMG LabTech to validate the compatibility of the Thunder technology with their multimodal plate readers. The first reader selected for validation was the CLARIOstar Plus. As for any TR-FRET assay, it is essential to set up the instrument correctly in order to generate the best results. The energy source as well as the proper selection of settings, including filters and other components, can all impact the instrument's sensitivity for any given assay. The CLARIOstar uses as a light source a high-energy xenon flash lamp. Regarding the filters, we use the HTRF TR-FRET filter set. This set is based on europium and a far-red fluorophore like the one used in Thunder. TR-FRET assays should be measured in dual emission mode, detecting both the emission from the acceptor fluorophore at 665 nanometers and the europium donor at 620 or 615. This allows to process data using the ratiometric method. In addition, the europium signal is very helpful when troubleshooting. For the other instrument settings, including the delay and counting time, we use values that have been shown to work well with a variety of other readers. We also wanted to verify whether settings recommended for alternative TR-FRET technologies were appropriate for the Thunder technology. Therefore, assay plates were also read using their corresponding instrument settings. Lastly, we compare results obtained with the CLARIOstar with those obtained with Envision, which is the plate reader we use in our lab for reading TR-FRET assays. For the Envision, which also has a xenon flash lamp, we use the settings optimized for Thunder. The first assays tested on the CLARIOstar were the phospho-protein assays. All phospho-protein assays are based in a sandwich immunoassay format. The assay workflow consists of 3 steps. Following cell treatment, cells are lysed with the specific lysis buffer provided in the kit. Then, the specific phosphoprotein released in the cell lysate is detected with a pair of fluorophore-labeled antibodies in a single-step reagent addition, no wash steps. We conduct the assays manually using the 2-plate assay protocol where the cells are cultured, treated, and lysed in a 96-well culture plate, and then the lysate is transferred to a low-volume 384 well plate, followed by addition of the labeled antibodies. Here we show the results of a quality control assay used for releasing the phospho-4-EBP1 assay kit. 840-31 cells were cultured in a T175 flask. Following lysis, lysates were tested neat and serially diluted in the lysis buffer. The same plate containing the lysate samples was read on both the Envision and the CLARIOstar. It can be seen that the signal-to-background ratio obtained with the neat lysates versus the buffer alone is higher with the CLARIOstar than with the Envision. In addition, with the CLARIOstar, there were no major difference among the different TR-FRET settings tested. It is interesting to note here that the lysate titration data show that the FOFO4-EBP1 assay has a wide dynamic range, despite the fact that the lysate was generated at very high concentration in a T flask. This figure shows a concentration response curve for the FOFO-ERK1-2 protein. These data were generated using HEK293 cells treated with serial dilutions of EGF. Again, the same detection plate was read on the ENVISION and the CLARIOstar. It's clear that the CLARIOstar provides a higher dynamic range compared with ENVISION. Regarding pharmacology and signal-to-background ratios, both readers provide comparable data. Again, no difference between the TR-FRET settings tested were observed with the CLARIOstar. The next phosphoprotein assay tested was phospho-P38 alpha beta gamma. Results are summarized in the figure on the left. HeLa cells were incubated with serial dilutions of anisomycin to stimulate phosphorylation of P38, and the detection plate was read on both readers. While pharmacology was comparable between the readers and settings, a higher dynamic range and signal-to-background ratios were obtained with the CLARIOstar versus the ENVISION. In parallel, and using the same batch of cells, we generated cell lysates and tested phospho-P38 protein using the reagents included in the SysBio assay kit. In this case, the detection plate contained two concentration response curves, one generated with the Thunder kit and the other with the SysBio kit. That plate was read on the CLARIOstar only. These results are summarized in the figure and table on the right. It can be seen that while comparable pharmacology was obtained with both kits, the Thunder kit generated a higher signal-to-background ratio versus the SysBio kit. The second step of the validation work with the CLARIOstar consisted in testing a panel of cytokine assays. Thunder cytokine assays are based in a sandwich immunoassay format and are conducted in two steps. Following cell treatment, cytokines are released into the culture medium. Then the supernatant sample is added to a 384-well assay plate, and the cytokine is detected with a pair of fluorophore labeled antibodies in a single-step reagent addition. For each validation experiment, we run manually standard curve assays in 384-well plates using recombinant proteins diluted in assay buffer. The figure on the left shows a typical nonlinear standard curve for the human EL1 beta assay. Each data point is the mean of triplicate wells plus minus one standard deviation. A single standard curve was run in the 384-well plate, and the plate was read on the ENVISION and the CLARIOstar. Data were fit using the 4-parameter logistic curve, and then the EC50, SB ratio, lowest detection limit, and lowest quantification limit were calculated. Those values are summarized in the table below the figure. The first thing one notes is that the CLARIOstar generates data with a higher dynamic range compared to the ENVISION. EC50 values were comparable between readers. However, the CLARIOstar generated higher SB ratios. In addition, the CLARIOstar generated approximately 5-fold lower LDL values compared to the ENVISION. The higher sensitivity obtained with the CLARIOstar is more evident in the figure shown on the right. This figure shows a zoomed view of the low concentration range of the standard curve shown on the left. This linear plot gives a much clearer presentation of the signal increase within this part of the standard curve. It shows that for the CLARIOstar, there is a systematic increase in signal over the low concentration range. This, combined with the very low interwell variation, make it possible to discriminate between these low concentrations. Here we show results obtained for the human EL12 assay. The figure on the left shows a typical nonlinear standard curve. Again, each data point is the mean of triplicate wells plus minus one standard deviation. Data was fed using the 4-parameter logistic curve to calculate EC50, SB ratio, LDL, and LQL. Those values are summarized in the table below the figure. For these assays, we measure the plate on the Envision using either the flash lamp or the laser, and we compare the data versus the CLARIOstar. The CLARIOstar generated data with a higher dynamic range compared to the Envision, even when using the laser for excitation. EC50 values were comparable between readers, and the highest SB ratio was obtained with the Envision when using the laser excitation. However, the CLARIOstar generated approximately 10-fold lower LDL values compared to the Envision using the lamp, and also lower LDL values compared to the laser. The much higher sensitivity obtained with the CLARIOstar is more evident in the figure shown on the right, which shows a linear plot of the low concentration part of the standard curve shown on the left. This plot shows that for the CLARIOstar, there is a clear and systematic increase in signal over the low concentration range. With Envision, the increase in signal at the lowest concentration was not only for the laser. The last assay tested was the human TNF-alpha. The standard curve for this assay is shown on the left. In this case, the standard curve was measured on the CLARIOstar using 3 different TR-FRET settings and also on the Envision. As was the case with the previous assays, the CLARIOstar generated data with a higher dynamic range compared to the Envision. The EC50 values were comparable between readers, but the CLARIOstar gave higher SB ratios. Again, the CLARIOstar generated approximately 10 times lower LDL values compared to the Envision, and this regardless of the TR-FRET settings used. In fact, in our hands, the TR-FRET settings used had little, if any, impact on the performance of the assays. The figure shown on the right shows a linear plot of the low concentration part of the standard curve shown on the left. This plot clearly shows the much higher sensitivity afforded by the CLARIOstar reader. In conclusion, we have shown that Thunder is a versatile and cost-effective TR-FRET assay technology platform particularly suitable for cell-based assays. These assays are all homogeneous and require a single reagent addition. They enable easy, rapid, sensitive, and robust quantification of endogenous proteins in either cell lysates or supernatants. The validation of selected Thunder assays read on the CLARIOstar Plus showed that in our hands, these readers generated a higher dynamic range and comparable or superior SB ratios to the Envision for phospho-protein assays. The CLARIOstar also generated lower LDL values, that is, higher sensitivity, than the Envision for cytokine assays. This finding might open new assay applications using lower sample volumes. Lastly, we confirm the Thunder assays do not require special filters and settings. The Thunder assays work optimally with CLARIOstar filters and settings recommended for other TR-FRET technologies. Overall, Thunder assays combined with the CLARIOstar Plus provide a compelling alternative to ELISA and other TR-FRET technologies. We are looking forward to validating additional plate readers from BMG LabTech. If you have any questions or need additional information, please visit our website or email us. Thank you very much for your attention.
Thank you, Jaime. That was some really excellent data, and it really encourages us at BMG to pursue further collaboration with BioAuxilium, and I'm really excited to see going forward the performance of our other readers, especially the Pherastar FSX. So the Pherastar FSX is our top-of-the-line, high-throughput screening microplate reader. And so it is designed to be both fast and extremely sensitive in all detection modes. And that really comes to light with the detection in time-resolved fluorescence. And that's depicted here with this LOD comparison, where you can see that the Pherastar FSX in 384 well plates, there is no microplate reader with better sensitivity. So Jaime also indicated the need or desire to miniaturize assays, and that is, of course, something that we think about with the Pherastar FSX as well. And as you can see here, when the limit of detection is miniaturized to a 1536 well plate, again, no other reader is more sensitive than the Pherastar FSX. So there are a number of things that contribute to the performance of the Pherastar FSX, but it really starts with the optic module system, which is depicted on the right-hand side of your screen. Now these optic modules have all of the filters as well as dichroics, everything that is necessary for high performance in all detection modes, and they are assay specific. So you just swap one in and swap one out and are able to instantly have the capability to read a new type of assay. Another thing that contributes to the Pherastar's performance is the fact that it has a UV laser that is dedicated for use in time-resolved fluorescence. This provides both higher excitation energy, but also better speed. As you can also see from the optic module system, these incorporate the ability to have two emissions at once, or simultaneous dual emission. And this is really important when you're trying to improve read speeds for assays like TR-FRET. Another thing that is really important for time-resolved fluorescence and TR-FRET is that not only do we have two PMTs, but we have dedicated matched PMTs specifically for time-resolved fluorescence. So we really have put a lot of thought into making giving you the best performance in the microplate reader. Thank you again for your time, and we hope that you will look into BMG LabTech and BioAuxilium. Thank you.